Welcome everyone. Good morning and welcome to this Water Institute WaterTalk at the University of Waterloo, co-branded this time with the Waterloo Institute for Nanotechnology. My name is Roy Brouwer. I'm the Executive Director of the Water Institute. And I want to start by acknowledging that we are participating here today from traditional territories of the first people across the country. Here in the Waterloo region, I'm on the Haldimand Tract, land that was promised to the Haudenosaunee of the Six Nations of the Grand River. And here you see a map of the land. The Haldimand Tract is within the traditional territory of the neutral Anishinaabe and Haudenosaunee peoples. As you can see from the map here, there is a discrepancy between what was promised and where the original inhabitants of this area live, and what was ultimately given as part of the agreement. The reserve is on approximately 19,000 hectares, whereas the original size of the land was almost 400,000 hectares. I encourage you all to take a moment to recognize the traditional land wherever you are. Before I introduce today's speaker, just a few housekeeping items. Please add your questions to the Q&A box, not the chat box. And you can do this at any time throughout the talk. And my Water Institute colleague, Nancy Goucher, and Lisa Pokrachak from the Waterloo Institute of Nanotechnology will then help to get you to these questions at the end of the Water Talk. Only use the chat box, please, for general comments or technical issues. And this webinar will, as always, be recorded and posted to the Water Institute's YouTube channel afterwards. So at this point, it's my pleasure to introduce the speaker for today, Juliette Legler. Dr. Juliette Legler is Chair of Toxicology at the Institute for Risk Assessment Sciences at Utrecht University in the Netherlands, where she also heads the Department of Population Health Sciences at the Faculty of Veterinary Medicine. Previously, she worked at Brunel University in London and Vrije Universiteit Amsterdam, where she became a full professor in 2013. We were colleagues for many years in Amsterdam. She developed an award-winning grad course on ecotoxicology, in which I also gave a guest lecture on economics of hazardous chemicals. Juliette is also no stranger to Waterloo. She got her undergrad degree in environmental studies from Waterloo and her master's and PhD degrees in environmental toxicology from Wageningen University in the Netherlands. Her research focuses on understanding how exposure to endocrine disrupting chemicals and other contaminants such as microplastics may affect health. And that's what she's going to talk about today. Juliette has led various international research projects and currently coordinates the European Commission funded project Goliath, an acronym for the generation of novel, integrated and internationally harmonized approaches for testing metabolism disrupting compounds, and Momentum, a microplastics and human health consortium funded by the Netherlands Organization for Health Research and Development. In her talk today, Juliette will focus on the relationship between microplastics and health, as I mentioned. And Juliette, we're very happy to have you here with us today. I'm happy to open the virtual floor to you.
Thank you very much, Roy. It's a real pleasure to be here. Let me share my screen with you. And let's take a look at my presentation here. Hopefully it will go into slide mode. Can you see that okay?
Yes.
Great. Well, thank you again for this lovely introduction. And it's really such a pleasure to be here, especially considering that Waterloo is my alma mater, and I have many fond memories of special times at the University of Waterloo. Today I'll talk about microplastics and human health, so not so much about the endocrine disrupting chemicals. I'll talk about this new field and how we can move the field of microplastics and human health forward. Just a small aside, indeed, I studied in Waterloo. I realized this was about 30 years ago that I studied there. I don't know where the time has gone, but I had such a wonderful time. And I did the co-op program, and it was actually at a co-op work term in the Netherlands where I first got to know the Netherlands and first started my work on environmental toxicology. And when I went back to Waterloo to finish my degree, I spent about 9 months in the lab of George Dixon in the aquatic toxicology lab, and that research led to my first paper back in 1997, published with Paul Sibley and George Dixon. Wonderful memories for me, and really an honor to be invited to speak to you all today. So I just wanted to briefly, before I get into the science, talk a little bit about my university that I'm at now, and I'm really hoping that this talk will be an opening for collaboration with you all, and that one day soon, when these crazy times are over, we can visit each other again, and I hope that you'll be able to come and see our beautiful campus here in Utrecht, in the center of the Netherlands, one of the biggest universities in the Netherlands, and a university that's very diverse with over 30,000 students, 7,000 staff members, and established over 450 years ago. So a long-standing tradition of academic excellence. Very, very proud to be working there. Our university focuses on 4 different main research themes, as you can see here. Our mission is a better future for everyone. The 4 themes include dynamics of youth, institutions of open sciences, life sciences, and pathways to sustainability. And within the pathways to sustainability theme, our plastics research across the whole campus of the university is expanding and developing. So if you would go to our website here and look for our U-Plastics 3, Utrecht Plastic Sources, Sinks, and Solutions Network, you can see that we're a very interdisciplinary group of researchers working on plastics and microplastics, from oceanographers to chemists to physicists to biologists, toxicologists like myself, but also health scientists and law, governance, and policy people. We're looking to expand this network outside of Utrecht, so I hope again that this talk will be a call to you all to collaborate and to look at possibilities for working together in the future. So my talk this afternoon for me and morning for you will focus on micro and nanoplastics. So I'm sure that many of you are familiar with this group of contaminants. In case you aren't, I thought I would do just a few slides of background. So micro and nanoplastics, well, the definition of microplastics are small plastic fragments less than 5 millimeters, and nanoplastics referring to these plastic particles less than 100 nanometers. Here's a picture in the right-hand corner of my screen here of some examples of micro and nanoplastics, and just to illustrate their diversity in shapes and sizes, in colors, and that we're talking about a very complex group of contaminants. This slide also shows that microplastics are made up of many different polymers, so the building blocks of the particles. In addition, many additives are added to plastics. So we have to take into account not only the particle and the plastic polymer building block of the particle, but also all of the additives that have been added to the plastic to give it its certain property. But also that the morphology can be quite different. It can be round polystyrene beads, where most of us have done our research on up till now. In real life, in the environment, we'll find many different kinds of plastics in different shapes, fibers and fragments and spheres, and films and foams of different colors. And in addition to having additives added to the plastics, once they're in the environment, can also absorb different chemicals as well, which are called ecotoxins here in this particular figure that I borrowed from this paper. So we'll get back to that in a moment. So very complex group of contaminants again. So plastics are made up of polymers. Polymers are the main ingredients. You can see here some examples of the most widely used polymers and then their uses in many different products, from water bottles to baby bottles to plastics to cleaning products and various kinds of food packaging. As I mentioned, micro and nanoplastics are not only made up of plastic polymers, they're also made up of plastic additives and all sorts of processing aids and different chemicals to give the plastic its specific the shape or size or quality characteristic. This is an interesting paper that came out very recently that shows that there are over 10,000 different unique chemicals can be found within plastics, including additives and different processing aids. Interesting in this paper is that about a third of the the chemicals that have been found are actually uncategorizable, so unidentified or uncategorized plastics that we really don't know, or additives that we really don't know their source and their identity. This figure also shows what kind of classes of chemicals we're talking about here, from metals to organic chemicals, but also where the major uses are for these kinds of additives in these plastics, their production volumes. In many cases, we're talking about plastics that are used in huge production volumes per year, and it's a first glimpse into their potential toxicity, so that we know that they may be endocrine disrupting chemicals, EDCs, or they may be known to have aquatic toxicity, or they have carcinogenic, mutagenic, or reprotoxic qualities. So again, a complex group of contaminants, plastics that are particles and additives. And this small figure here is indicating, again, that not only the plastic additives within the plastic may be of concern, but also the chemical contaminants in the environment, for example, in the aquatic environment, that may sorb to the plastic debris and become part of the microplastic and ultimately be available for uptake to the organism. So when we talk about microplastics, we have to keep in mind that we are again talking about huge volumes of plastics that are produced on a yearly basis, on a yearly level, and these plastics will break down into smaller plastic particles and ultimately become micro and nanoplastics. And so in this slide, I'm indicating, the huge extent of this plastic problem, with 8.3 billion tons produced since the 1950s, and estimates showing that only a small portion of these plastics are actually being recycled. Majority is either incinerated, or by far the majority is disposed of in our landfills or in our natural environment, including our aquatic environments, as litter. And some estimates have calculated that about 12 billion metric tons of plastics will be in landfills worldwide by the year 2050. So an ever-increasing problem and an ever-increasing exposure to plastics in our environment. Of course, plastics, as I mentioned already, will break down. They tend to break down slowly. It depends, of course, on the plastic type. There are different kinds of degradation processes. This can be thermal degradation, hydrolytic degradation, photodegradation. There's also some biodegradation of plastic. There are some micro organisms that can break down some sources of plastic, but in general, this is a very slow process. And ultimately, the hope is that the plastics will break down into inert or non-dangerous substances like carbon dioxide and water. However, the evidence now is that this will take many, many years, and until the plastic actually degrades completely, we will have smaller and smaller and smaller particles being formed, and in which our environment is exposed to, but also ourselves. So where are the major emissions from plastics coming from? So this is a study that was done in the Netherlands recently, and I don't think it would be much different in Canada. So as I mentioned, the major source of plastics is land-based litter. So not surprisingly, land-based litter fragmentation is a major source of microplastics, microplastic emissions, reaching in a small country like the Netherlands already 10,000 tons per year. Other important sources of microplastics include the wear and tear of our tires, from our automobiles and our vehicles. Paints are consisting of microplastics. Pellets, pre-production pellets, are actually intentionally made small plastic particles. We also have plastic particles that we use in cosmetics and in our cleaning agents. The washing of synthetic fabrics is an important source for microplastics as well, as well as rubber infill and artificial turf fibers. So multiple sources and multiple potential exposure sources for humans. So when we talk about understanding the risks of micro and nanoplastics, so what is the actual potential health risk to humans or to organisms in the environment, we have to think about the exposure on the one hand and the ability of the microplastic to bioaccumulate in an organism. And on the other hand, we need to think about the potential hazard of the nano and microparticles, so the potential effect on our biological systems. The thing about microplastics is that they're not only complex in their size and their shape and their source, their type of plastic, they're also very complex in their toxicity. This makes them a really interesting group of contaminants to study. So when we think about microplastics as toxicologists, we think about three different kinds of effects. The one effect is the actual particle toxicity itself, and we know that particles can cause toxicity, they can cause immune reactions, they can cause oxidative stress, different biological responses in that they are a particle themselves and can damage membranes and enter cells. The other factor that's important in understanding the toxicity and the potential health effects of plastics is that they have the potential chemical effects, given these wide numbers of chemicals that are actually added to plastics or sorbed to plastics once they're in the environment. And the third and really important and increasingly studied potential effect of microplastics is their potency to sorb microorganisms on the outside of the particles, and in that way to act as vectors for pathogens that are coming from microorganisms, from viruses. And so the transport of nasty pathogens through these particles is a really important field that's burgeoning at the moment, is getting a lot more attention, and certainly in these COVID-19 times. So up to now, most of the work done on understanding and trying to understand the effects of micro and nanoplastics has been done in laboratory studies with model organisms, model species that model mainly the environment. So fish and aquatic invertebrates, plant species, and sometimes in higher vertebrates as well. So on over 100 species, there have been studies done, there are publications available that are showing that microplastics, when dosed in controlled laboratory environments, can lead to effects at different levels of biological organization. So changes in molecular and cellular effects, changes in systemic effects, and effects on apical endpoints, which we would call then an adverse effect, like changes in locomotion and growth, reproduction, and feeding of the species. The thing with most of these experimental studies is that they have been done in using mostly high exposure concentrations, so not environmentally relevant exposure concentration. This is because in the lab we want to know, what is the actual level at which these particles can cause toxicity, and generally we start those kinds of exposures by using high doses. Another caveat of this work up till now is that it's not easy to obtain microplastic particles, and certainly not easy to obtain microplastic particles that are relevant for the exposure of organisms in the environment. So most of the studies done up till now would be have been done with a specific type of plastic that is commercially available, that a nice round, for example, the round polystyrene beads are one of the most widely tested plastics up till now. And these are not really the plastics that we are using in the field, or that we are exposed to in the field or in the environment or in our everyday lives. So this means that we have limited field evidence, and absolutely no human studies have been done with microplastics, as far as I know, up till now. So this has led myself and my colleague Dick Fatak to put together an overview of some of the knowledge gaps that are needed to ascertain human health risks of microplastics. We published this work earlier this year, and we provided this commentary on really on the one hand, to indicate how much we know about species in the environment and how much we know about macro litter and the effects on fish and birds, but how little we actually know of human health effects. And so it's really important to do more research there. We summarize some of the major knowledge gaps. As you can see on this slide here, when it comes to human exposure, there are very few robust and accurate and reproducible analysis methods for measuring micro and nanoplastics in environmental samples, much less in human samples. So the studies looking at micro nanoplastics in human blood or in human tissue are very, very rare at the moment. Luckily, there's a lot of research ongoing to improve that. But until we have these good reproducible methods that can also measure very small nanoplastics, we really don't have a good idea of internal exposure. And these are very challenging studies and challenging techniques. So in addition to lack of knowledge on human, in particular, internal exposure, we have a lack of knowledge on whether these particles are actually taken up and what determines whether the particle is taken up, and if it's actually causing toxicity or if it's just excreted. As we know, we can measure micro nanoplastics in human feces, so that gives already the indication that a lot of plastics are eliminated from our body. The question is, which plastics are not and where do they end up? So this is very much a factor of their uptake, size, source, and shape, whether or not there is a specific coating we call the corona or the biocorona around the plastic particle, and how these plastics are absorbed and distributed and metabolized and ultimately excreted in the body. I mentioned already the importance of these three different types of toxicity. There's still a lot of knowledge gaps when it comes to human models and looking at these toxicity. And ultimately, we really don't have a reliable method for dealing with the risk. There are no risk assessment frameworks yet for micro and nanoplastics. There are for nanoparticles. I think we can learn a lot from the nanotechnology field. I think we can learn a lot from what we know from air pollution as well when it comes to assessing the risk of micro and nanoplastics. So the Dutch government and Roy mentioned already the Netherlands Organization for Health Research has been quite a trendsetter, I think, in terms of medical research funding internationally, in recognizing the need for more knowledge on human health effects of micro nanoplastics. And this is quite unique from a perspective of a medical funding agency that tends to fund medical research. They're actually funding more and more environmental research or environmental risk factors in human disease, micro and nanoplastic exposure being one of them. So this funding agency came out 2 years ago with funding for so-called breakthrough projects, in which short-term research was funded, high-risk research, 15 projects were funded in order to provide some of the first steps that were needed to better understand human health effects of micro and nanoplastics. So in my own group, we were lucky to have been awarded 3 of these breakthrough projects. One of the projects was focusing on whether or not microplastics are taken up by the intestines using a human intestinal organoids and understanding human immune responses in the intestine to microplastics. The second project in my group was carried out by a neurotoxicologist, Remco Westerink, who studies whether or not plastics can be transported over the blood-brain barrier and cause effects on the developing neurological system. The third project was looking at what happens when microplastics, and do microplastics make it to our human fetal environment, and if so, what potential toxicity can they cause there? So for that third project, I'd like to go into a little bit more detail. I've included the website of these breakthrough projects where you can find the results of these projects. And later on in my talk, I'll talk about the follow-up research from this big initiative to fund 15 breakthrough projects in the Netherlands. So coming back to the question about microplastics in the fetal environment, in the environment of the developing baby. So when we started this research, there was no research yet, no publications yet on the uptake or presence of micro and nanoplastics in the human fetal environment. So we considered this a high-risk project because we actually didn't have any evidence that micro and nanoplastics were actually taken up. We based our hypotheses that they are taken up by two other lines of evidence. First of all, this line of evidence that air pollution particles have been demonstrated in placentas. So you see this figure on the right here in which black carbon particles of nanometer size have been demonstrated in the fetal side of the human placenta, demonstrating that indeed the fetal side of the human placenta can take up these particles from the maternal bloodstream, and then this can potentially cause health effects. We don't know yet, but we know that air pollution is associated with a number of negative health outcomes for the mother and the child. The other line of evidence that led us to believe that micro nanoplastics would be taken up in the human placenta is coming from animal studies. So this is one example of an animal study in which fluorescent microplastics were injected into pregnant rats, and the distribution of the fluorescent particles through the body of the pregnant rodents and into the developing offspring within the uterus of the rat were examined. And in this study, it was clear that these fluorescent particles, which are shown here as these little green balls, were visualized and found not only in the placenta, but also in the fetal brain and the fetal lung and the fetal liver, indicating that they can also be taken up by the developing offspring. In the meantime, since we've been carrying out this research, this summer, this interesting publication came out from a group in Italy, confirming that microplastics are found in placentas. This was a study in which human placentas were collected at delivery, and using imaging techniques, some different microplastic particles were actually imaged in the placenta and confirmed that there is indeed uptake of microplastics in the human placenta. So in our study, we wanted to also confirm, do microplastics reach the human fetal placenta? Can they cause harm to the placenta? The placenta being this really important temporary tissue in during development of the baby. It's really important for providing nutrients to the developing baby, providing oxygen to the baby, for metabolism, for example, metabolism of hormones between the mother and the child, hormones that are really important for the development of the baby. So any effects on the placenta could have effects on the developing child. So it's a really important tissue to focus on. And we know for many years now that the placenta is not a very good barrier to chemicals. We know that many chemicals taken up by the mother will be transferred to the baby through the placenta. And we expect that might be the case for microplastics as well. So we studied not only the placenta, but we also looked at amniotic fluid, the fluid surrounding the baby during development, for the presence of microplastics. We were also interested in whether the chemicals associated with microplastics could cause harm. So the way to go about this, of course, is no longer doing this work by yourself in your laboratory, but working together in multidisciplinary teams. So we put together this fantastic team of scientists in order to address these questions. And we were very appreciative of the collaboration with a pediatrician who provided us with the placenta samples and amniotic fluid samples. We worked together with a great team of chemists from Amsterdam and also from a partner from the US. And these are the partners who are working so hard on developing sensitive and robust and reproducible methods to analyze micro nanoplastics in human placenta. We worked together with environmental scientists who provided us with very environmentally relevant samples of microplastics that we actually weathered in the laboratory, so we could look at the difference between pristine plastics and weathered plastics. And we work together with environmental epidemiologists to start to set up the first human epidemiology cohorts and studies in humans. And we work together with cell biologists who have the best imaging and microscopes possible to image these tiny little nanometer-sized particles, which are actually, similar sizes to some of the actual organelles within a cell. So the first work we've done is to develop two different methods to measure microplastics in human tissue. One of the methods is working with pyrolysis GCMS. This was done in the Netherlands in the Amsterdam lab. This is a destructive technique in which the sample is extracted and really destroyed through pyrolysis, and what is left are the plastic polymer fingerprints that can be measured with the GCMS. We also work together with chemists in the USA working on high resolution MS in order to identify these plastic polymer fingerprints in the samples. This allows us to identify the actual concentration of the polymers, to identify the polymer, but it doesn't allow us to say how many particles were in the placenta or the size of the particles, because they're both destructive techniques. At the moment, we're working on new methods in which we combine the pyrolysis GCMS with high resolution microscopy methods that would allow us to, in the samples where we find the plastics, to zoom in and look under electron microscopes and other types of microscopy and imaging techniques to actually detect the particles and count them. So with these methods, we've actually have very strong indications for plastics in placenta and in amniotic fluid. And this work is being published as we speak, so I can't show you any actual data from the work, other to let you know that we have very strong indications that they are indeed there in both amniotic fluid and in placenta. So in addition to actually showing that these plastic nanoparticles are in placenta and in the fetal environment, we also want to understand better the potential hazardous or toxic effects in the human placenta. Well, to do this, we've been culturing placenta in a petri dish. We use different human placenta cell models, and we use a high resolution microscopy to actually image the uptake of these microplastics. So in this little film here, what you see are placenta cells in a petri dish that are being imaged by a confocal microscopy. What you see here in yellow are the proteins in the cell. The blue are big round nucleus of the cell, so you can tell that these are individual cells. And what we've done is we take images through this monolayer of cells, and what you can see is in the 200 nanometer, the very small particles. It's not working. Oh, and Julie asked me if I had any images or slides, and I said no, but I've actually forgotten that I wanted to show this one. Doesn't matter. I have another slide to show it. That's this one here. This is a similar image of the film that I just wanted to show, and what it's showing is these small green particles are actually the fluorescent polystyrene beads, the microplastics in the placenta cells. We've tested them at different sizes, so 200 nanometers, 1 micrometer, 10 micrometers, and we can confirm that these plastics are actually taken right up into the cell. They're not just sitting on top; they're within the cell, and all three sizes, from these relatively big plastic particles to the very small ones, are taken up very, very efficiently by both types of placenta cells that we've looked at. And the importance of the difference between these cells is that these cells here below, the syncytiotrophoblasts, are actually the really endocrine active cells that are really important for producing hormones and metabolizing hormones for the baby during development. So we've confirmed that these polystyrene beads are taken up We've done work with weathered particles and can confirm that they are taken up. And we've done research on looking at, so what happens in the cell when these particles are taken up. We've been looking at both the gene expression events and the metabolites that are being expressed in these cells, and can confirm that there are definitely changes to these gene expression and metabolomics effects. However, this work is also, I need to publish this. We're publishing it very soon, and once it's been rigorously reviewed, I'll be happy to show some of the data here. But we have the first evidence that particles are taken up and that they're changing biological reactions in terms of gene expression and metabolites in the cells. We cannot answer the question yet if they cause harm to the placenta yet, because we need to be looking at longer term exposures and the long-term effects of these changes to gene expression. We don't yet know if the chemicals associated with microplastics cause harm. Again, there we are looking at trying to identify these chemicals because we have indications that there is more effect when you expose the cells to a pristine particle than to a weathered particle. And this has likely to do with the chemicals that are still in the pristine particles that are causing the effects. So these studies need to be verified in larger sample sizes and larger populations, understanding long-term effects and understanding toxicity, not only of the polystyrene beads, but of many different plastics with different sizes and shapes. So in the last few minutes of my talk, I'd like to talk about how we're going to go about researching this potential health effects of these micro and nanoplastics. And we're doing this within another large project that our medical funding agency has funded, the so-called Momentum project. Momentum stands for Microplastics and Human Health Consortium. It's a project that started in June of this year. It's a really exciting project in that it brings together all of the investigators of the 15 breakthrough projects that I mentioned earlier, together with private sector participants in order to work together on plastics research, but also to work on plastic solutions and solutions to these projects, to these problems. So we're representatives from universities, research organizations, medical centers, small, medium enterprises, and trade organizations and large enterprises, including some of the large plastics producers in Europe and internationally. The project will run for 3 years and represents over 5 million euros of investment. I'll come back to these partners in a moment. So the idea is to really build on the momentum gained in these breakthrough projects and move towards a long-term research infrastructure on micro and nanoplastics and health. So this project runs for 3 years, but our goal is to really develop a long-term 10 to 20-year research theme on micro nanoplastics and health and solutions. So the project focuses on many of those research gaps that I identified in the introduction to my lecture. So better understanding how micro nanoplastics are formed, characterizing internal human exposure, characterizing the immunological hazards. We found in the breakthrough projects that in almost all of the human models that were studied, given the lung models, intestinal models, placenta models, the response of the immune system was really the first and foremost response that we need to better understand. We need to develop a risk assessment roadmap, and we need to develop solutions and future research agenda of a public-private partnership. We need to work together on solving these problems. So this is the structure of the Momentum project. It's on the one hand based on fundamental research on analyzing the problem, and on the other hand works on coming up with solutions, including risk assessment and other solutions, working together with industry for minimalizing potential health risks. So I'll just briefly run through the different parts of the project again, this is an opening and a call for collaboration with you all. I think you might have noticed on one of the earlier slides, we're always looking for new partners in this project as well. So the first work package in the project focuses on formation and characterization. It also generates reference micro and nanoplastics for the toxicity studies. And it also, really importantly, these reference micro and nanoplastics often need to be labeled to have some sort of fluorescent label or some other dye so that we can actually visualize them in our cell models. And we will also be generating more weathered particles so we can better understand how plastics change when they travel in the environment and how that affects their toxicity. When it comes to exposure assessment, again, a lot of this research will be focusing on internal exposure, these robust methods for measuring micro nanoplastics in human blood and in human tissues, and in order to set up the first human biomonitoring studies, which we'll be doing in this project. It's also really focusing on the translocation of particles over important internal barriers of the lung and the intestine and the placenta and the brain, and better understanding the role of the corona, the film surrounding the microplastics on the uptake and toxicity of these particles. When it comes to hazard assessment, again, as I mentioned, focusing on immunological hazards in these various human tissues, considering that lung and intestine are the most important exposure routes in inhalation, and food being really important exposure routes of micro nanoplastics. And here we also have the study on pathogens and what is the role of pathogens in the immune response, and what is the persistence of these pathogens on micro nanoplastics in our human body and in our environment. Finally, the project will develop a risk assessment roadmap, major steps needed for risk assessment. We have policy makers from the Dutch government on board, and we will work also on solutions. How can we co-create solutions with private sector governments, NGOs, and private citizen representatives? And when it comes to solutions, these are some of the solutions we are examining and developing long-term roadmaps for, including new and alternative materials, safe by design. Recycling is an important solution route that our industrial partners are working on. But also, how do we link human health assessment to environmental health assessment, where there's been a lot of work done in the last 10 years? And how do we set up large-scale human health evaluations? And it's all moving towards a public-private partnership for long-term research in this field. So I just wanted to conclude by showing the logos of all of the different public and private partners involved in this project, and thanking them very much. We had our first research meeting recently when we, in the Netherlands, were allowed to have meetings again. Unfortunately, this type of meeting is not allowed anymore, given the COVID measures and the numbers in our country again. But it sure was fantastic to see everyone in person again. And as a final note, I also wanted to note that in Europe, a lot of research money has recently been dedicated to microplastics and human health. 5 large research projects have been granted that have started in April of this year within this CUSP cluster. You can see the website here. So again, more opportunities for micro and nanoplastic research in human health, but also more opportunities to collaborate and to work together on this huge challenge. So thank you all very much for your attention. And I'll stop sharing and go back to the moderators. I see that it's gotten very dark in my room here, and I'll turn on my light.
Thank you so much, Juliette. That was really excellent. I learned a lot. It's a little scary as well. Before we dive into questions, our video producers would be able to show the animation on slide 25 if you'd like, or we can just jump right into questions. What's your preference?
I think we can dive into the questions. If you're really interested in that movie, if you Google my name and you look on YouTube, you can find me presenting that movie in another venue. So no problem. It'd be a pity to lose time from the Q&A. But I am going to turn on my lights in my room. I didn't, I'm looking so much at my screen. Just a moment, please.
No worries. There we go.
That might be better for visualization.
Okay, perfect. So the first question comes from Philippe van Capellen. So his question is, currently the World Health Organization maintains that there's no definitive evidence that micro or nanoplastics are harmful to human health. So do you anticipate this to change in the near future?
Yes, I do. I do, because first of all, when that study was done, there is very little research on micro and nanoplastics in human health. These projects are just starting up, so there will be a lot of research findings coming out in the next 5 years. What I also think is that because of the analytical challenges of actually detecting and measuring micro and nanoplastics, most of the studies up till now have reported some of the larger size microplastics. What we don't know and what we need to know better is what is the exposure to the smaller particles, and then related to that, what are the health effects? So that's really a big research gap that the World Health Organization study just could not tackle, because we just don't know yet.
Great. And I think I'll call on my colleague Lisa to ask the next question.
Yes, your talk was very enjoyable and very informative. And as Nancy said, it's a little too alarming. It is something that we thought was particularly inert in the atmosphere for air pollution in water. And I think I read a few articles, and a little bit disturbing on as an sort of academic, and to read something, let's say in The Economist, to say that, plastic pollution, it's not, it's a nuisance, but we shouldn't worry about it because it's particularly inert. And this was only a few years ago. To have a respected publication say something so bold and not recognize that, well, we think it's inert just because there's fewer studies on it. I think it needs to be revisited. Anyways, I am happy to represent the Waterloo Institute for Nanotechnology, and taking over from Nancy, who's done a great job. We have a question here. Our audience member asks, I'd be interested to hear a bit more about the challenges regarding the procedure to get access to pregnant women to allow them to do tests to see if microplastics are present in fetal waters. Would you be able to explain a bit more how this works in practice, and how do you collaborate with hospitals on this?
Oh, that's a great question. Well, it certainly helped in our case that we have a very proactive pediatrician on the project who's already very concerned about exposures to chemicals and exposures to plastics in his own patients. So he's been very proactive in helping us here. So in the Netherlands, and I can imagine it would be the same in Canada, we just can't take placenta and amniotic fluid samples. We have to set up a study. We have to have that very clear study protocol, how we want to do that. We have a special committee that of medical experts and other experts who would then look at our protocol and test it if it's indeed medically and ethically acceptable. And we need to have that approval before we could even start a study like this. So that's the first important thing. Once we had that approval, what I understand from my colleague pediatrician was that it was absolutely no problem to find volunteers. First of all, the placenta and the amniotic fluid are both very easy matrices to get samples of at delivery. There's the big placenta is usually discarded So that's not a problem, and the amniotic fluid can be sampled if possible at the delivery, if it's a hospital delivery. So he was able to get these samples for us. I should say the challenges in the actual analysis of micro nanoplastics also resides in the plastics are everywhere problem that we have, and that all of the sampling material and the containers that are used needed to be tested beforehand by our chemists to make sure that we had the best type of containers that would not give us contamination in our analysis, because everything's plastic, right? And we also have to make sure that we include the right controls in the whole analysis procedure in order to distinguish what might have been introduced during the sampling and storage of the samples and what's actually in the samples during pregnancy, right? And that's why the chemists in my projects are very cautious about publishing their work and making sure that they have all of the right checks and balances to really definitively say that what they're measuring is actually inside the sample and hasn't been introduced in some other way, or even in the lab, which is also full of plastic. So yeah, quite a challenge there, but you can get around that by working together with a good group, and with a good chemistry group. Our paper should be coming out soon on measuring in human blood and placenta, and then you can see what kind of plastic containers or glass containers, if possible, can be used for sampling the placenta and the amniotic fluid, and all of the necessary checks and balances to go from getting your samples to actually determining if there's micro and nanoplastics in them. So I hope that answers the question. Okay, and I'm sure that our pediatrician would be happy to share insights with whoever asked this question. So just send me an email, and I'll be happy to bring you in contact with them.
Thank you. We have another question. Is there any fingerprinting involved also to see where the microplastics come from that they detect in the amniotic or in the fluid? So I think he's meaning amniotic fluid.
Yeah.
Is this possible to also warn pregnant women what products to perhaps avoid?
Well, that's another really good question. As far as I know, there's no way yet of determining where the microplastics have come from. Of course, we can determine what plastic type they are. So also, if it's PET or PVC or polypropylene, but where it comes from, I think that's a really good question and a real big challenge. I imagine that if we have these fingerprints, we may one day be able to say a certain fingerprint is indicative of a certain product, but I'm afraid it's too early for that. As far as I know, that's not yet possible. What I can think of as well is that our research or global research into the corona, the film surrounding the microplastic, might also give us hints as to where the plastic has been. And the second question regarding prevention. That's of course the reason why we're so solutions oriented. Even though we don't have the answers, and I know it sounds all very alarming what we're saying, but we still don't really know if it's harmful to humans because we don't have the exposure information. We don't have the toxicology information yet. Until you are able to say at the current exposure levels, there are adverse effects. Until we can say that, I don't want to be too alarming yet. However, we know that we're exposed, and the exposure to microplastics will only increase given the huge amount of plastic and the increase in plastic. So we also already want to work on solutions, even if it isn't such a big problem to our health. And that's where we think that working together with industry on these recycling initiatives, on safe by design, so designing better plastics will help. And there are, of course, a number of NGO initiatives, really good initiatives to better track your your plastic use. The Plastic Soup Foundation, for example, has an app available where it can help people become more aware of where they're exposed to plastics and where they may be able to avoid exposures. For me, the most important thing is that we work on big scale global solutions to close the plastic circle, to make this a circular use of plastics, and also to have the correct or robust risk assessment procedure in place in order to really make the policies that we need as governments to assess the risk and determine what measures need to be taken if there is a risk. So not only the onus on the individual pregnant woman, but really the onus on the policies and the large-scale initiatives that are necessary.
Thanks. Really well said, Juliette. Question from Nastran. Can microplastics change DNA or RNA? If yes, how does microplastics change DNA, since they are particles and not chemical components?
Yeah, really good question again. So there are some indications that microplastics can be genotoxic, so toxic to the DNA. That can be through binding to the DNA because some of these plastics are reactive. I don't think it's known if that's because of the particle itself or because of the chemicals in the particles that cause this genotoxicity. I'm not an expert in the field of genotoxicity, but I'm pretty sure we don't know yet how exactly that works. In our own studies, we didn't actually examine effects on the DNA itself. We examined the effects on expression of DNA, and what we saw is that genes involved in the metabolism of hormones. This is a really important process in the placenta. Those genes were changed. Again, it doesn't mean that it's harmful. It doesn't mean that it can't be recovered after a certain amount of time, but it's the first indication that the cell is reacting differently to the presence of these particles in the cell, and these enzymes, these genes that code for enzymes that are so important for metabolizing hormones, for the hormones that are needed for development of babies. These genes are changing. So we really need to know what that means. And that's the first indication to me that something's happening in the cell. Whether or not that's going to turn long-term harmful, I hope not. But that's really what we need to understand better.
Okay, I with the presence of Roy, Roy, you're on mute, by the way. We have a few more questions, but we are coming to time. Thank you so much for all of your sharing your wisdom, insight, knowledge, and letting us know the state of the future steps for human health and micro nanoplastics in the environment.
Thank you.